more valuable teaching time? Okay, translation. For those of you who are hungry, don't worry, I'll let you go a little early so you can eat before the exam. I'm already hearing people say that they're hungry. Okay. Whoa, what happened?
That's what we're trying. So tiny.
But that's better, I don't have the ears. Who's blind in the back and can't see? Who, take off your glasses and tell me if you, oh wait, no, you can wear your glasses. Okay. Does anyone have a hard time seeing this? I think it's not that bad, but I'm up here. And my husband calls me Eagle Eye. That's a pet name to me. Okay. So, I have good eyes, what can I say? Exam this week, if you haven't figured that out, that's bad. Our final exam, can you believe it? Hey, hi, how you doing? Hey. Okay. Final exam, we're having a final exam. Final exam is coming right up also, on the 20th. And it's in this room at 10:15. So don't get confused, because I've had people walk in at 11 before, and then you miss like half the time from your exam. Which really doesn't make a difference for us, because it's an hour-long exam, but still. But it's here, in this room. So those of you people at home listening, it's not in your bedroom. It's in our lecture hall. Okay. So this is where we ended the other day. We've been talking about translation, and I was going through the major steps. And it's just basically addition of the new tRNA to the A site. Then you get the formation of your peptide bond, and then your large ribosomal subunit moves one way, and then that results in movement of, and then the small ribosomal movement, the small ribosomal subunit moves, and then that results in movement of the tRNAs into all over. So you've got your number 3 in the E site where it can exit, because E stands for exit, and you have a vacancy in the A site where your new tRNA comes back in, and that just repeats over and over again. And the peptide bond is catalyzed by peptidyltransferase activity in the large ribosomal subunit. Actually, I feel like I found something inconsistent in the textbook, because I wrote this down: peptide bond formation catalyzed by peptidyltransferase in large ribosomal subunit. That came out of the textbook, and then I was reading in preparation for today, and it said how the ribosome is mostly made of RNA, and there are some proteins, but those proteins are mostly involved in stabilizing the RNA structure, and that in reality, the peptide bond is formed by more the RNA, not the protein. So now I'm confused, so don't worry about that detail until I work it out. Out of the same textbook, I don't know. Anyways. Because it's a ribosome. So, we're talking about translation. Let's talk about how this actually happens, okay? So you've got elongation factors that are required for 2 things. One is to improve efficiency, so it's helping to bring in the tRNA to the ribosome, and to assure accuracy. It's helping to make sure the correct tRNA amino acid is present. So there are two different elongation factors involved in this: EFTU and EFG. In bacteria, and EF1 and EF2 in eukaryotes. EF for elongation factor. So if on the exam I say anything about EF something, you already automatically know it has to do with translation elongation. And so EFTU, it actually brings in the tRNA to the ribosome. So you can see it's this little moon thing bound to the tRNA, bound to that amino acid, and helps to bring it to the A site. And actually, this is all GTP dependent, and GTP hydrolysis that happens upon correct binding allows that EFTU to go away, and allows then you get formation of your peptide bond. So if the tRNA is not fitting correctly, then it's going to be in one conformation where it's more able to leave the site. So it checks the accuracy of the codon-anticodon match, so that's one thing, because it's binding to, checks accuracy of two things, the amino acid tRNA, so it's binding to the amino acid and the tRNA, so it's checking that match, and then it's also checking the codon-anticodon match, and these are all very important things, right, because you don't want the wrong amino acid added to your protein. That's pretty important. So I want you guys to start thinking about what mechanisms the cell has in place to ensure appropriate addition of the right amino acid that's input. Because what's the point of having a genome if random amino acids are going to be added, right? In all reality. Okay. Right? Should we just have ribosomes going wild, just binding things, making peptide bonds? Okay. Oops. So what was I just saying? EFTU, so in bacteria, this is EFTU, it's called something else. Eukaryotic cells, it escorts the tRNA to the A site, so there you see it in big font and big pictures. EFTU binding to the tRNA amino acid, and it checks for accuracy. And it's involved with removal of the incorrect tRNAs. So an incorrectly base pair tRNA will dissociate. And so if you have correct base pairing, so it's actually kind of, so let's say you have correct codon-anticodon base pairing, then you're going to have GTP hydrolysis, which will allow for conformational change in the EFTU and then dissociation. Yes?
Does each tRNA carry only one amino acid?
Yes. Fantastic. Thank you. I love these comments. Does each tRNA carry one amino acid? That tells me, that question tells me that there might be confusion about what the tRNA is doing. And so let's just talk about that for a minute. Right, so tRNA has a codon, an anticodon at one end that recognizes a codon, and at the other end binds to an amino acid. And that's how you bring in the appropriate amino acid per codon. You have your mRNA that you've made that's got all these nucleotides, and every three are a codon, right? That encodes a certain amino acid that's going to be added. And so each tRNA binds to a codon, and so that's what's happening here. tRNA comes in, binds, the anticodon recognizes the codon, and then if it's a correct match, then you get the peptide bond forming between the two amino acids, and the tRNA goes away and can become recharged with another amino acid. Did I somehow touch upon anything that may have been confusing? Yes?
So how does like the RNA, like they said there was one that helps tRNA go into it?
Yes.
How does it do that?
So there's a lot of ribosomal RNA involved. But one thing which I'm sure you're probably talking about is this one, probably, the 16S ribosomal RNA. So tell me if I'm answering what you're thinking of. So this codon-anticodon matching is very important, and it is kind of proofread, in a way, by the 16S ribosomal RNA, so a portion of the ribosome. And what it does is, here's your anticodon, and here's not the whole thing, because at the molecular level, there's this hydrogen bonding that's happening between your anticodon and your codon, and also your 16S ribosomal RNA. And if it's binding correctly, then it's going to stimulate GTP hydrolysis and EFTU release. Does that make sense?
Yeah.
It's kind of cool that it's actually a part of the ribosome that's also helping to check the accuracy. Also, incorrect tRNA molecules can bind weakly to a codon, but they take longer for the peptide bond to be formed. So that gives it another opportunity for those tRNAs to leave if, for whatever reason, EFTU leaves, even though it's the wrong tRNA. So you think about proofreading a little bit in this. So proofreading is happening through the action of the EFTU. It's happening through the action of the 16S ribosomal RNA, and it's also happening at some earlier stages that we talked about, right? So I want you to think about all these different mechanisms we've been talking about that are used to make sure that the appropriate amino acid is added. So who can think of another mechanism? Another something we learned about that's ensuring the appropriate amino acid is going to be added to the growth polypeptide chain. Do you guys want to talk to your friends about it for a minute? Discuss? Okay, I see some people nodding. So why don't you guys talk for a minute and think about it. We just talked about the activity of EFTU and bringing in and making sure the right tRNA amino acid is brought in. But there are other points in which the wrong amino acid is being avoided from being added. I don't even know what language I'm. Talk for a minute.
Oh, yeah, one. I think
Okay, thank
Okay, so we'll talk. So I hear some discussion about something that might be right, but we're having a hard time remembering names of things. So who can tell me how else the cell makes sure the right amino acid is added to the protein at a stage earlier than what we just talked about? Let me see. Let me look. It's not even worth looking at my list because no one's here. Let's see. Nope, not here. Sean.
It would be one mechanism is the different tRNAs coming in and binding to each codon separately, and it's not just one enzyme doing that, it's the tRNAs coming in.
Okay, so something about the tRNAs coming in separately. So each tRNA needs to come in and bind to the codon, right? Okay, so that's one thing. So who else can think of something else? Yeah.
Would be amino acid transferase now.
Yeah. So the tRNA synthetase. So what adds the enzyme that we talked about at length. No, well, now he's making me doubt my naming of the enzyme. I thought tRNA synthetase, aminoacyl tRNA synthetase.
Okay.
So the enzyme that we talked about at length on Monday, that adds the amino acid to the tRNA. Remember, it has an editing site, it has to add correctly. So that right there is a huge mechanism for ensuring the right amino acid is added because it makes sure the right amino acid is added to the tRNA, right? So one aspect is the correct amino acid added, and then I guess it's kind of one I was dividing into two, but yeah. So ensuring the correct amino acid is added to the tRNAs. One, ensuring the correct tRNA is brought into, ensuring the correct tRNA is brought into the ribosome is two. And then you can think the regulation of the binding, the tight binding, right? So EFTU is doing one part, but then how tightly the interaction between the codon and anticodon are, that's kind of another one, right? Because if it's not strong, it will detach. Anyone else? Anyone want to add? So 4, it's more like 3, because I was kind of thinking of the aminoacyl tRNA synthetase as 2 mechanisms in a way. Yeah.
Are the EFTs, are they different depending on the amino acid they bring in?
Are the EFTs different depending upon the tRNA they bring in? Well, they do bind to the amino acid and the tRNA, so they are somehow checking the accuracy there. So, which begs the question, but I don't know. I'm not sure. Yes.
Could chaperones help?
Did chaperones help with what? What? Proofreading. Well, it depends on what kind of chaperone you're thinking of. What kind of chaperone are you thinking of? What would it be doing? Because there are chaperones that help with protein folding, right? Like HSP 70, HSP 60.
I can't remember the name, but there was one that, when as soon as the ribosome synthesizes the protein, it was working right next to it.
HSP 70, it binds to every single protein coming off the ribosome. That's probably what you're thinking of. But then you already have protein. So it's not going to really help with bringing in the tRNA. So that's like the next step, which if you take 408, you learn about the which maybe that's where you learned it with some. But yeah, pretty much in terms of thinking about amino-acyl tRNA synthetase, you do want to think about the two mechanisms within there, because one is whether or not the amino acid can fit in the binding site, and the other is whether it fits or not in the proofreading site. So it kind of has two mechanisms within that, which is how I came up with before. So just think about that. That's a nice categorizing question, right? It's a good essay. Okay, so let's think about the ribosome. It is 2/3 RNA. So it's mostly made out of RNA and about 1/3 protein, which is pretty amazing, right? Yes?
Are RNAs and tRNAs post-transcriptional
Are ribosomal RNAs and tRNAs post-transcriptionally processed? Which kind of processing are you thinking of?
just any
Any, are they spliced? No. Are they capped? Are they polyadenylated? No. I think that some of these can have some cleavages and some modification, which I really didn't get to teach you too much about, so kinda. But in the,
There'd probably be some mechanism to make sure they fold, right? Or would they do that by themselves?
So the comment I just got, is there some way of making sure they fold correctly? Do they do that by themselves? Well, a lot of folding is spontaneous. There might be some RNA, I don't know. I don't know if there's an RNA chaperone that helps in folding. But these foldings are based on the sequence, and they're going to fold into whatever's the lowest energy structure. Just like proteins, actually. It's just that proteins are a little bit sloppier.
But I mean, sometimes you have to go through steps with proteins in order to like cosylation and all this to make it happen.
Yeah, but this, none of those modifications are happening. Yeah, I mean, I guess you'll have some, well, let's not go there. We're going to get off topic. So I would say processing in the mRNA sense, no. But yes, there's sometimes certain cleavages and stuff like that. Okay, any other questions? Any questions at all? Okay, so you can see this is all RNA, right? So all this purple, all these things, this is all RNA, and you've just got these little proteins stuck in there, and it's mostly for structure. And so this is showing it more nicely. It's all RNA except for some of these ribosomal proteins that kind of stick on the outside and help to hold things together. Pretty amazing. So they're ribozymes, which means they're RNA with catalytic activity. And that, what is that catalytic activity that they have? So they catalyze. How about Kelsey? Sure. You're just a Kelsey to me. I'm sorry.
Oh. Well, there's two other options.
I know, but I'm looking at you. I'm gonna go with you.
So ribosomes catalyze the formation of proteins.
Okay, and more specifically, peptide bonds. Yep, absolutely. So they're ribozymes. And this is where I was reading this, and I was like, wait, they're saying, it's okay. So we talked about elongation actually before we talked about initiation. Does this bother anyone? It's not very chronological. It actually bothers me, but that's what they covered it in the book, so I kept it in that order. But let's go back for a minute to initiation. Okay, so you actually need to assemble. You need to do two main things to initiate, let's say. You need to find your RNA, assemble your ribosome on top, and that'll make that 3 things. Bring in your first tRNA and find your first AUG, right? So you have your small ribosomal subunit. It'll bind to the initiator tRNA, and that's brought in by another factor called EIF2, initiation factor 2. Let's go. I got this more specifically here. So the initiation factor binds brings in the tRNA to the P site and assembles with the small ribosomal subunit, which then the mRNA is brought in, and that all happens through interaction with the cap. See the cap? Remember the cap? It was important, right? Not just for RNA stability, but for interaction with the ribosome. So the ribosomal subunit is going to bind to the mRNA by recognizing the 5' cap, and then you start scanning for the first AUG. And it's not in here, but you should be thinking to yourself, wow, how is this small ribosomal subunit making it through this RNA that's full of secondary structure? There's actually other initiation factors. I'm sure you guys are all thinking that, right? There are other initiation factors which have helicase activity. So if I ask you if helicase activity is required for translation, you might be able to say yes. And that helps the ribosome move through the secondary structure in the RNA. Okay, so there's so remember, when you make your RNA, it forms this loop structure. You've got your poly A tail here, and so you actually have EIF4E, which binds to the 5' cap, and then EIF4G that binds to the cap and the poly A tail. That brings it into that conformation. And then you have recognition of the 5' cap by the small ribosomal subunit, and then scanning for the first codon, the AUG. So then the AUG is found. Yay! Are we excited? Yay, AUG. The EIFs dissociate, and the large ribosomal subunit is brought in, and it binds, right? So a few things to think about. How is the correct AUG identified, right? If you look in it, remember, how many bases are in RNA? How many kinds of bases? 500? No. 4 bases. So what are the odds that you're going to find an AUG? Right? There's a ton of chances of finding an AUG. So ribosome needs to identify the correct AUG. You need to find an AUG that's an appropriate reading frame, right, to make the protein that we want. This is important. So there's actually a consensus recognition sequence. Whenever you are thinking about specificity of how and when things happen, I always think there must be a consensus sequence. And so there's actually this sequence, ACC, AUGG, which is called the Kozak sequence, and is a source of an argument that I had with, actually it wasn't really me, it was more of a coworker of mine and my boss. During my PhD, we were trying to express a gene in a plasmid, and he was like, well, is there a Kozak sequence? Because it wasn't expressing it at all. Like, we cloned it in, we had a clone, we had a sequence, everything looked perfect, we transfected into ourselves, nothing. And so he's like, well, did you put a Kozak sequence in? And we're like, huh? Which is probably how you'd all be, because how much time do you spend thinking about Kozak sequences? What, like 5 minutes that we're spending right now? And we didn't put it in, so the protein didn't express, and the experiment didn't work, and then we had to go back to cloning, it's like a lot of time to clone something, and then have to go back because you stupidly designed your primers poorly. So that's why I teach you about Kozak sequence, so that when your boss says to you, did you put in Kozak sequence? You say, yes, of course I did. Thank you, Dr. Ehrlich. So there's a sequence that is directly upstream from the AUG, which helps, it's actually part of where the AUG is. Here's the AUG, ACC, AUGG. If your AUG is in that context, then you're going to be recognized by the small ribosomal subunit and your initiator tRNA. If your AUG is not in this context, then the ribosome subunit's going to ignore it and move on to the next AUG. Yes?
Is there, if there are more than one AUGs in the reading frame, is it still going to add a methionine?
Yes, methionine could be, methionine is not only the beginning of a protein. Methionine is anytime an AUG comes up. Yeah. So the question was, if there are more AUGs in the reading frame, can you add a methionine? Yeah, why not? Yes.
Consensus sequences, like, is it that when you get a consensus sequence, even if it's in DNA or RNA, so if it's in RNA, that means it's going to be the same consensus sequence in DNA?
So you're asking what the general definition of what a consensus sequence is?
Yes.
Yeah. So a consensus sequence is just a sequence that's frequently the same in the same situation. So, for example, when we talked about splicing, there's a consensus sequence on your 5' splice site and your branch point and your 3' splice site, all splice sites, all the time, everywhere, and it's because it's recognized by certain splicing factors that participate in the splicing reaction. There's this consensus sequence, has nothing to do with that. This consensus sequence is always the same where the first AUG is in eukaryotic cells, and that helps the small ribosomal subunit when it's scanning for the first AUG to know that that's the first place to start translation. There are consensus sequences for every transcription factor. Every transcription factor has a specific consensus sequence, which is a sequence, in this case in the DNA, that the transcription factor wants to bind to initiate transcription. But still, it's like the same sequence might be present in a million different genes, but that's what's recognized by the transcription factor. This sequence is what's recognized by the small ribosomal subunit plus the initiator tRNA.
So you can have it for like amino acids too.
Yeah, absolutely. So we have, in the past, we were trying to understand how different proteins interact. And we looked through many different proteins of many types that have similar domains, and you're going to find maybe there's a consensus sequence for an interaction point between two different proteins. So it really means a sequence that's the same, so there's consensus. If we all voted, and we all voted for something, there'd be a consensus, right? We'd all be in agreement. So a consensus sequence is a sequence that's in agreement among multiple genes or whatever you're talking about to have some function. Does that make sense? Yes?
But the AUGG is the only real conserved region, or the only real conserved sequence, and that the ACC is an indeterminate significance. Is that what we should know?
I'm never going to ask you to repeat back a consensus sequence to me. I will ask you to understand the function of a consensus sequence in a certain context. So if I ask you, how does the ribosome know where to start translation, you would say, well, we'll look for the Kozak sequence or the AUG in the context of a consensus sequence that is important for a ribosome binding site. It's a consensus sequence, which means that it's not always ACC, AUGG, but there are variations to that. So let's talk, so this is, you're scanning for your AUG. So there's a concept called leaky scanning, and that's where sometimes you'll get translation initiation and sometimes you won't. Sometimes it'll skip and go to the next methionine, and that's used purposely sometimes by the cell to produce different proteins with different N-terminal sequences. And this is useful, for example, if you're trying to produce a protein that is secreted or membrane bound, right? Or that is sent to the ER or sent to mitochondria. Remember, there are signal sequences that determine where proteins are going to go. Well, don't remember because I haven't told you unless you took short weight, but hopefully you've learned somewhere there are signal sequences that determine where proteins are going to go, and you can choose whether or not you want to have your signal sequence or not you. Like, it's not like you're like, no signal sequence of protein A. No. But there is that flexibility, and it's produced by leaky scanning, which is sometimes you'll start at one AUG, sometimes at another. Yeah. What regulates leaky scanning? I don't know, but I'm sure it's regulated. Right? It's got to be regulated. So if you want something, an easy one is if you're gonna have something secreted or membrane bound, let's say. Secreted has not membrane bound, secreted or going to some other place, because signal sequences are fairly short. And if you skip over your signal sequence and don't have it, it'll be secreted out of the cell. My guess is I've learned this before.
If a gene has more than one functional reading frame, wouldn't it stand to reason that the Kozak sequence would agree less with the consensus, so that it binds tightly to one specific place?
Yeah. Okay. So wait right there.
Okay.
Wait right there. So one thing is what determines which one's going to be made, right? And so yes, binding tightly is going to determine if that AUG is going to be recognized or not. But what determines whether or not it's going to bind tightly? That I'm not 100% off the top of my head, but yes, there are mechanisms for regulating that. And it likely has something to do with some factor that we haven't learned about. Should we make that an extra credit? It's been a while. Who wants an extra credit during an exam week? Well, whatever, you don't have to do it. Let's do it, okay? So the question is, what regulates leaky skin, right? So what determines when you're going to have one AUG recognized or another AUG recognized? If you think about it in terms of alternative splicing, we talked about it in terms of there are these repressors that are going to kind of bind in high splice sites. That's going to be a little bit different here, I think. But when you hide your strong splice site, then the cryptic splice site is recognized. And so one would guess it's something similar, but I don't know. Yeah.
So in essence, you're saying that for one mRNA, you can get several proteins from that?
From one mRNA, you can get several proteins.
Well, from a mature.
Yeah. Yep, I am saying from one mature mRNA, you can get several proteins. Not just through this mechanism, there are many mechanisms. You can get RNA editing that's going to produce multiple different proteins. And I believe that's what controls secreted or membrane-bound antibody. Okay. Any other questions? Pretty cool, huh? Everyone get that extra credit? It's recorded. I'll try to get that off early today. Okay, good? Happy? Okay, so let's just go through the process. You'll have your amino acyl tRNA that binds to the A site, right? So you've already scanned. You found your right AUG. Your initiator tRNA is going to bind to the A site. And then you're going to have your other one, your second amino acid tRNA, bind to your P site, A site, and then you have the formation of your peptide bond between the methionine and amino acid number 1. And actually, it goes on and on and on to what we were talking about before with elongation. You keep having that movement of your ribosome so that then you have, once that peptide bond is formed, this one is free to leave, right? And everything moves over. So this moves over, and then the A site is empty again to accept the next tRNA. And it goes on and on and on until termination. So let's bring up another thing that I was thinking about. Bacterial mRNAs aren't capped, right? Does everyone agree? Who thinks they're capped? Okay, so that was gonna have to fail you. Okay. Bacterial mRNAs aren't capped. So how are they recognized by the ribosome, right? How does translation happen with them? So they contain a consensus sequence, and as you can see here, the true meaning of a consensus sequence, right? There's some variation. There's some variation in the distance between the initiation codon and the Shine-Dalgarno sequence, but they're all about 10 nucleotides or so upstream, and they all contain generally this sequence, and that is a ribosomal entry site for bacterial RNAs. Okay, so you don't have the recognition of the cap like you do with eukaryotic cells. Okay, and so if you think about bacterial RNAs, remember bacterial genes sometimes are organized into operons, and they'll be controlled by the same regulatory region, which means you'll get this long RNA with many different genes, right? Polycistronic, 1, 2, 3, and there'll be multiple AUGs, and then there'll be multiple of these Shine-Dalgarno sequences that are the ribosome binding sites. So you can get an entry of your ribosome even in the middle of your RNA and translation of the individual proteins. Okay? So that's a nice compare and contrast. Think about the difference between how things work in terms of initiation in bacteria versus eukaryotic cells. Any questions? Okay, so then we have release factors, right? So we started, actually we've translated, then we started. So we started translation, we elongated our polypeptide, and now we need to be finished, right? And that happens when we encounter stop codon, right? Do you guys all know your stop codons? It's okay. So basically, once you encounter stop codon, there's no stop codon recognized by a tRNA. It's recognized by a release factor. And so you'll get this UAG, which is one of the stop codons, and it'll bind to a release factor. And then you get addition of a water molecule, so there's your OH. And since the only thing that's holding your protein to your ribosome is interaction with your tRNA, your protein is released, and then your ribosome disassembles. So here are your stop codons in case you felt like knowing them: UAA, UAG, UGA. So binding of release factor, addition of water molecule, release of protein. So the protein and mRNA are released and the ribosome disassembles, and it can be used again. So release factors are an example of molecular mimicry. And molecular mimicry is exactly what it sounds like. It's when a molecule mimics the function of another molecule. And in this case, here's your tRNA. That's what a tRNA looks like. And the structure of a release factor is similar to that of a tRNA and allows it to bind to the A site of the ribosome and terminate translation. So it makes sense that they bind to the same place, they should have a similar structure. And so don't forget, just like you can have multiple RNA polymerases making multiple RNAs on one gene, you have multiple ribosomes that translate the mRNA at the same time, and that's part of the reason that this loop structure is so efficient, right? So you find this AUG, you get translation, 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 then you're done, right? Your release factor binds, you release your protein, you disassemble. Oh, look, you're right near the start site. Let's hop back on and make more protein, right? Multiple proteins from one RNA because multiple ribosomes combine and translate one after the other. And then to add a level of awesomeness, so this all happens in, where in the cell does this happen? The cytoplasm. And in bacteria, there is no nucleus, right? So actually, this is happening at the same time that transcription is happening in bacteria. Because as soon as the RNA is coming out of RNA polymerase, ribosomes can hop right on and start translating. Very efficient. Yes?
So you have multiple proteins being translated at the same time, and there can be different proteins. You said
Oh, because of the scanning? Is that what you're connecting? So you could have multiple proteins translating at the same time, and they could technically, why not, be different proteins? Because if there's some, depends on what's regulating the process. Okay? It depends on what's regulating. And so that's what we need to know. Because if there is some factor that's obscuring, for some reason, the first one or causing skipping of the first one, then you would think that that could affect everything for the period of time that that factor is bound to the RNA, right? If it's a randomness, which is also weird, things don't just happen randomly often in the cell, that seems not to make sense, then you could say multiple proteins, multiple different proteins off of the same RNA.
so we're hypothesizing the factor associated with the RNA not ribosome that causes the disease.
Well, I don't know. We don't know. It's hard. I mean, until we know that, we can't even think about it, right? Right. So if you think about it, if it's something that's binding to the ribosome, then yes, right? Because maybe it's just controlling the activity of that one ribosome. If something binding to the RNA, then it's going to control the output from that RNA. Right. Yeah.
Are there different types of ribosomes?
Are there different types of ribosomes in eukaryotic cells?
Yeah.
I don't think so. Well, tell me where you're coming from with that thought.
Oh, I'm just wondering because like other things seem to be like specific to certain sequences and stuff like that. So I was wondering if maybe a ribosome might be like specific to a certain protein or something like
So
that.
if there's like a level of regulation at the ribosome assembly level. I have not that I know of, but that doesn't mean that there is. Yeah. Good thinking. Because I guess once you've regulated transcription and you've got all that RNA, then
Sorry.
I mean, there's so much regulation at the level of whether or not your gene's going to be transcribed and whether or not your RNA is going to survive. But yeah, it's something to think about.
Necessary.
Well, no, I mean, you never know. It could just not be known yet. That might be your future Nobel Prize. Okay, so that's it for now. Go and try to grab a snack before the exam and you're taking the exam.